Hello. I'd like to thank Galena and the organizers for this opportunity to present at this conference. I'm sorry that I cannot be with you today to present in person. In this talk, I'd like to explore the impact that PAX6 mutations have on the brain and endocrine system. I do not have any conflicts of interest to disclose. In addition to vision, people with PAX6-mediated aniridia report symptoms and have signs associated with changes in the function of different sensory systems, including a reduced sense of smell, an altered ability to process sound, sensitivity to touch, and problems with the sleep-wake cycle. Individuals with aniridia can also exhibit ptosis, ataxia, and altered cognition typically associated with autism spectrum disorder. These observations raise the following two questions. First, do people with aniridia experience more of these neurological symptoms than do people that don't have aniridia? And second, can these neurological symptoms be explained by reduced PAX6 activity? In my opinion, these questions highlight a gap in our knowledge about aniridia, and that gap in knowledge can be framed by asking, what is the prevalence of neurological symptoms in people with aniridia? On this slide, I have the results of a pilot self-report study that my colleague and I conducted in the spring of this year. Although we are still analyzing the data, I wanted to share with you our preliminary findings. Interestingly, people with aniridia receive a clinical diagnosis of auditory processing disorder, autism spectrum disorder, and obsessive-compulsive disorder more frequently than was observed in the population of the United States as a whole. These results are shown in bold. In contrast, people with aniridia receive a clinical diagnosis of sensory integration or processing disorder, attention deficit or hyperactivity disorder, anxiety, and depression at frequencies comparable to that of the general population. The other preliminary findings of note are shown here. Individuals with aniridia tend to exhibit problems with motor control, shown in the left panel, and balance, shown in the right panel, more frequently than those without aniridia. I want to emphasize that these results are based on self-reporting of clinical diagnoses and are therefore subject to the biases inherent to these types of studies. Clearly, more careful study needs to be done to validate any of the preliminary findings that I have shared with you today. Nonetheless, it appears that individuals with aniridia do face some neurological challenges in addition to those associated with the eye. Conceptually, there are two ways that changes in PAX6 levels can explain changes in neural function. One mechanism is through changes in the way the brain is organized during fetal development, shown on the left, and the second mechanism is through changes in the ways that neurons function because of reduced PAX6 levels, shown on the right. To illustrate the first model, I've drawn a tissue composed of alternating gray and yellow cell types with a regular arrangement. Under conditions of reduced PAX6, the yellow cell is missing and is replaced by a purple cell type, changing the structure of the tissue and its function. To illustrate the second model, I've drawn a tissue with blue cell adhesion molecules. Under conditions of reduced PAX6, the identity of the tissue is retained, but there are fewer blue cell adhesion molecules between the cells, presumably resulting in a tissue that is less resistant to mechanical forces. Importantly, as we think about potential effects of PAX6 mutations on brain function, it is useful to remember that PAX6 is a sequence-specific DNA-binding transcription factor that is expressed in multiple cell types in the developing and adult central nervous system. I've chosen to illustrate this point with these figures from publications from Vijay Tiwari's lab and Alish Svekel's lab. PAX6 can act as an activator or a repressor depending on the target, turning on specific genes or preventing them from being expressed respectively. The PAX6 gene encodes for 3 different protein isoforms, each of which binds to specific motifs in the chromatin. As shown in this figure from Alish Svekel's lab, The PAX6 protein can bind to many places in the cellular chromatin. Some targets are tissue-specific, as can be seen in the numbers of binding sites specifically associated with the lens, cortex, or beta cells, and others are common to multiple tissues, as can be seen by the overlapping regions of the Venn diagram. Not surprisingly, the PAX6 protein controls gene regulatory networks involved in several different cellular processes. There's a lot of detail in these two figures on this slide, and I apologize for that. But what I want to draw your attention to are the cellular functions regulated by PAX6. PAX6 controls gene networks that control neural sulfate specification, neuronal differentiation, and axon guidance, as well as the cell cycle, protein trafficking, and metabolism. Therefore, changes in PAX6 levels have the potential to affect multiple aspects of neurophysiology. Here's one example of the changes in cellular physiology associated with a reduction in PAX6 protein levels. This data is from PAX6 deficient corneal epithelial cells. As you can see in this heat map, PAX6 haploinsufficiency alters the expression of specific genes in these cells. Shown here are the results from two biological replicates. Red indicates genes that are more highly expressed in one genotype compared to the other. The genes affected are independently associated with eye problems such as corneal stromal opacities, glaucoma, and myopia, as well as neurological problems such as cognitive impairment, among others. The use of RNA-seq and similar approaches can be used to identify PAX6-dependent pathways important for understanding aniridia. There is compelling evidence for structural and functional changes in the brains of individuals with PAX6-mediated aniridia, as is shown in this summary slide. Changes include a reduction in the olfactory bulb, the anterior commissure, the posterior commissure, the optic chiasm, the corpus callosum, and the pineal gland. In panel B, the image on the right is an example of an individual with aniridia with a reduction in the anterior commissure shown in the red circle, the posterior commissure shown in the blue circle indicated with the arrowhead to the left, and the pineal gland, also in the blue circle, indicated by the dotted arrow on the right. These are MRI images, and the right-hand side of the patient is to your left. In panel B here is an example of an individual with aniridia with alterations in the corpus callosum, indicated by the red box, and the optic chiasm, indicated by the blue box, again is visualized by MRI. In our study cohort, we did not see any statistically significant changes in the total corpus callosal area, mapped in red in panel A and shown quantitatively in panel B. Nor did we observe any changes in gray matter, shown in panel C, white matter, shown in panel D, or in total brain volume, shown in panel E, in people with PAX6 mutations compared to matched PAX6 normal controls. The structural changes observed in the adult human brain are likely due to changes in brain development. We know from studies in mice and other model organisms that PAX6 is expressed in different parts of the developing brain, as illustrated by this mouse embryo that I stained to show the cells that have PAX6 transcripts. These cells are labeled in blue. At this stage, we can see extensive expression in the developing eye, forebrain, nose, spinal cord, and in the endocrine pancreas. As illustrated by this cartoon, during cortical development, PAX6 is expressed in a rostral to caudal gradient with a boundary at the posterior commissure. This data, published by Manuel et al. In 2015 from David Price's lab, shows nicely PAX6 expression in the developing mouse brain. PAX6 is labeled in red. As you can see, PAX6 protein levels are highest in the rostral and lateral aspects of the developing cortex. As illustrated on this slide, PAX6 expression persists into adulthood in discrete structures and region of the brain. In this sagittal section to the adult mouse brain, we see that PAX6 is expressed by cells in the olfactory bulb, clusters of neurons in the cortex, in the hippocampus, several neuronal clusters in the thalamus and hypothalamus, and extensively in the cerebellum. Cells expressing PAX6 transcripts are labeled in blue. We can also visualize cells in the brain that express PAX6 using an expression transgene as shown here. Cells expressing PAX6 express the green fluorescent protein. As you can see, high levels of PAX6 expression are seen in the adult eye, cerebral cortex, and other brain structures. In addition to structural changes in the brain, there is good evidence that people with PAX6-mediated aniridia have functional changes in the brain. Bamiou first reported that persons with aniridia can have auditory interhemispheric transfer deficits and difficulty in localizing sound and speech under noisy conditions. In my lab, we tested this measuring electrical activity in the brain. We found that persons with aniridia have increased activity during early auditory stimulus recognition, likely subcortical and early cortical auditory processing, and decreased activity in late cortical integration responses. We think that this may represent cortical integration network differences between people with aniridia and those who are PAX6 normal, and also higher order processing changes in people with aniridia. Some of our data is shown here. In this test, we played 3 different tones to subjects who were either PAX6 normal or had a mutation in the PAX6 gene. We then measured the brain's electrical response. This response is innate and cannot be consciously controlled by the subject. As you can see, people with aniridia exhibited a larger electrical response to all 3 tones than did people who were PAX6 normal. This suggests that brains of people with aniridia are working harder to process auditory information. Consistent with this general idea, we found that persons with aniridia exhibit greater connectivity compared to PAX6 normal individuals when processing different tasks as measured by functional MRI. The parts of the brain exhibiting these changes are shown in green, red, and blue. Green is associated with executive control networks, red is associated with the primary visual system, and blue is associated with default mode intrinsic connectivity networks. The differences between someone with aniridia and a PAX6 normal individual in task-evoked brain activity suggest that individuals with aniridia recruit additional neural regions to supplement function in the critical intrinsic networks. I also want to highlight a difference in our imaging studies and those reported by Yonkara in 2016. The Yonkara study reported that people with aniridia exhibited a greater decline in the thickness of the frontal parietal cortex and an accelerated decline in working memory compared to PAX6 normal controls. We did not find either of these results in our studies. Clearly, more work is needed in this area. Turning now to the endocrine system, normal levels of PAX6 are required for proper development of the pineal gland, the pituitary gland, and neuroendocrine neurons in the hypothalamus. The pineal gland regulates circadian rhythms and is reduced in some individuals with aniridia. The pituitary gland controls various endocrine functions in the body. In our mouse model of aniridia, we found that mice with aniridia have increased numbers of thyroid-stimulating hormone-producing cells, here shown as TSH-producing cells, and increased levels of thyroid hormone in the blood. In the hypothalamus, neuroendocrine neurons secrete hormones that control other endocrine glands, including the pituitary, as well as those of several organ systems. We have found altered levels of hormone production in PAX6 deficient cultured cells. The physiological impact on people with aniridia is currently unknown. but preliminary evidence suggests that individuals with aniridia exhibit alterations in physiology attributable to changes in endocrine function. PAX6 is also expressed by cells that control metabolism. These include GLP-1 expressing L cells in the small intestine and cells in the endocrine pancreas. In both cell types, PAX6 is required for normal function in culture, especially in the context of diabetes. There are 7 main takeaways that I would like to leave you with. First, aniridia is more than just an eye disorder. Second, tissues that require PAX6 for normal development may exhibit altered development under conditions of PAX6 haploinsufficiency. Also, cells in adult tissues that express PAX6 can exhibit altered physiology due to changes in specific PAX6-mediated gene regulatory networks. Third, PAX6 is a sequence-specific DNA-binding transcription factor that is expressed in multiple cell types in the developing and adult central nervous system, as well as several types of endocrine cells. Fourth, some gene networks controlled by PAX6 are dosage sensitive. Fifth, people with aniridia have structural and functional alterations in the nervous system that include sensory systems, vision, smell, and hearing, as well as sleep-wake cycles and cognition. 6. People with aniridia may have changes in metabolism or endocrine function and appear to be at a higher risk for diabetes. 7. Understanding the gene networks controlled by PAX6 will aid in the development of pharmaceutical approaches for managing PAX6 deficiencies in adult tissues. In closing, I would like to thank the numerous people who worked on this project over the years, many of whom are listed here. I'd also like to thank our funders for the generous support that made this work possible. All of the individuals with aniridia and their families that participated in our research, and you for your kind attention. I'm happy to take questions via email.